Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installments in the ATCC Excellence in Research webinar series entitled The Protistology Collection at ATCC, Scientific Relevance and Best Practices, presented by Ms. Kathleen Glazer. Ms. Glazer is a senior biologist at ATCC. In this presentation, Ms. Glazer will discuss protistology, presenting what defines a protist and why these organisms are relevant not only to scientists, but also to the general community. She'll give an in-depth look at some best practices for propagating, preserving, and authenticating protists. Finally, Ms. Glazer will provide an examination of the special techniques that have been adapted at ATCC for the evaluation and cultivation of difficult-to-grow protists. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I would like to welcome Kathleen Glazer.
Thank you very much, Brian. Before I start, I'd like to say a few things about ATCC first. Founded in 1925, ATCC is a nonprofit organization with HQ in Manassas, Virginia, and an R&D and Services Center in Gaithersburg, Maryland. It is the world's largest, most diverse biological materials and information resource for microbes and is known as the gold standard in the industry. Today I'm going to be speaking about protists. We're going to cover a few different topics here. First of all, what is the definition of a protist? It's important to establish that first. Then we'll go over some examples of some protists and their relevance to biomedical research and the wider community. Then we'll briefly cover current and potential uses in industry. And then we'll get to the part of the presentation where I can really talk, techniques and applications, the things that I do every day. So first of all, what is a protist? Well, this definition is pretty important, so I'm going to go ahead and read it to you. Protist, any of a diverse taxonomic group, and especially a kingdom, Protista, synonym Protoctista, of eukaryotic organisms that are unicellular and sometimes colonial or less often multicellular, and that typically include the protozoans, most algae, and often some fungi such as slime molds. Now, that was a pretty long definition. Why does it need to be so long and involved? Well, the reason is because protistology as a field of study was initially created as sort of a catch-all group for organisms that didn't really fit anywhere else. You had your animals, you had your plants, you had your bacteria, and then you had these single-celled eukaryotes, and it was comfortable to call those protists for a while, but they're not all single cells, it turns out. So this definition is a little bit complicated. But that leads us into our next point, which is that this is a very diverse group. For example, encephalidozoan is a tiny parasite, 1 to 2 microns in length, and you're counting these things, it's really hard to see them. The other end of the spectrum, we have an amoeba called Bromia spherica, which can actually grow up to 1.5 inches in diameter. It lives at the bottom of the ocean. It's pretty cool. Now, the Protistology Collection at ATCC is the only general service collection of taxonomically diverse living stock protists in the United States, and the only large service repository of parasitic protozoa in the world. What this means is, suppose we have a customer in Hawaii who wants to order one of our bacterized amoebas. You would think that they would get it from someplace nearer to Hawaii, but they will actually order it from us, and we're all the way on the other side of the United States, the opposite side from Hawaii, and that's because we have this reliable and well-established collection. Some of the groups that we have include the amoebozoa, which is, have your amoebas, you've heard of that, the chrome alveolata with plasmodium, which is the malarial parasite, which is extremely significant, as we'll talk about later, and the epistoconta, which includes the encephalidozoan that I mentioned earlier. Now here we have a list of the highest demand cultures and reagents ATCC over the last 5 years that have to do with protistology. And there are a couple of things I'd like to point out about this list. First of all, in addition to the cultures offered on this list, or popular cultures, we also offer 4 different media, and even more significantly, I think, we have 9 DNAs on this list. DNA items have become very popular in recent years, and we do make a lot of DNAs with organisms such as Plasmodium that I already mentioned, Trichomonas, Entamoeba, and Blastocystis. Now, there are a number of clinically significant protists, so we have to spend a couple of minutes talking about this because they impact people's lives. They can cause human disease, and that is definitely significant. So let's go over a few of these examples. In the lower right here, we have Plasmodium falciparum, the malarial parasite, which sickens millions of people every year. It's a very big problem. In the upper left here, we have Toxoplasma gondii. Now, some estimates say that up to 60% of the world's population is actually asymptomatically infected with this. It doesn't cause any symptoms, so you wouldn't know it. And then we have, next to that, we have Trypanosoma cruzi that causes a disease called Chagas disease. It's very prevalent in South Central America, Mexico, places like that. And then next to that is Naegleria fowleri, which has been sensationalized in the media as the brain-eating amoeba. There's actually another brain-eating amoeba, Balamuthia, that we'll get to later. Then we have Trichomonas, Acanthamoeba, which causes an eye disease. Entamoeba and Giardia both cause dysentery-like diseases, gastrointestinal diseases. And then Leishmania, which causes a couple forms of disease, one of which is visceral leishmaniasis, which is in your gut, which is very dangerous. It's called Kala-Azar for the parts of the world, and also a skin disease. Now, I'd also like to talk for a moment, if I might, about some of the life cycles that some of these organisms have, because they're really kind of fascinating and involved. And as examples, I've chosen these 2. This one on the left here is the life cycle of Toxoplasma that I've already showed you a picture of in the last slide. This is a parasite that has a cat as an intermediate host. And you might have heard that pregnant women are not supposed to change the cat box. Well, this organism is the reason why. If a pregnant woman is infected with Toxoplasma, it can cause spontaneous abortion or birth defects, so it's very dangerous. So yes, the cat is the intermediate host, and then the cat eats mice and birds and things like that, and then it goes up the food chain and can eventually infect people and through blood products as well. But what's really fascinating about the Toxoplasma in particular is that it can actually sort of social engineer its own situation. So there is research to support the fact that the Toxoplasma parasite goes into the brain of the rodent, the mouse or the rat, and actually makes it run towards the cat instead of away from the cat as it would otherwise do instinctually. And it actually causes the cycle to perpetuate. So this is a very clever organism indeed. And then on the right here, we have, as another example, Cryptosporidium life cycle. And the reason I included this one just to show the complexity of some of these cycles. There's a meront stage, there's a gamont stage, there are a couple of different gamonts, and there's an oocyst stage. And then it's possible to be reinfected, and actually only one cyst is required from this organism to give you an infection. So it's pretty interesting. Now, I know it might look like I've been making protists look like the bad guy here. They cause disease, they do this bad thing, that bad thing. Well, I'd like you to know that they have a lot of scientific applications. Protists can be good, too. They're not all evil. So, first of all, they can be used in bioassays. As an example of that, they can be used for the detection and measurement of very small quantities of vitamins and growth factors. So, for example, Euglena and Opromonas can be used to estimate vitamin B12 concentrations down to 1 nanogram per mil, which is a very low level. So they're useful for that. They're also useful as molecular models. So if you look at that top slide there, that's a transgenic Trypanosoma cruzi with a green fluorescent protein molecule spliced in. GFP is taken from a bioluminescent jellyfish. And you might wonder, why do we do this? Why are we trying to give them superpowers? Well, we're not. We're just trying to use this as a reporter of expression. So there's another gene in there that you're interested in, and this is kind of a marker for that. But they just happen to look really cool, too, because they glow in the dark. And then as far as diagnostics go, there is a disease called lupus, which is an autoimmune disease. It's pretty serious. And there are some organisms that can help in the detection of autoantibodies against that points to the fact that you have lupus, like Cryptidia luciliae. And also, protists can be used as biofuels. They already are being used as such, and more research is ongoing as an alternative to fossil fuels. So biofuel production from protists is advantageous because, for one thing, these protists can survive on land that is not suitable for crop growth, so we couldn't use it for anything else. We're talking about algae now. Also, the lipid portion can be extracted and converted to biodiesel, and the carbohydrate content of an algal biomass can be fermented into bioethanol and biobutanol. It is a sustainable form of energy without diminishing the food supply, so it's a very promising field. And finally, they can be used for fatty acid production. You might have heard of DHA in commercials for baby formula because it's an important chemical to the development of the brain, which is largely fat. So Cryptocadinium and Aurontichytrium, Throstochytrium, Schizochytrium, and a few other organisms do produce DHA. So this can be used as a supplement for human health. Now let's talk for a moment about propagation of protists. We have to grow these in the lab, and there are a number of ways to do that depending on what we're growing. So the ATCC product sheet is the standard. There's one available for each ATCC item, and if you follow that, you should be doing pretty well with that. But there are a wide variety of techniques that I personally have used and have seen others in my lab use to grow these things. Just as an example, most protists should probably be thawed quickly at 35 or 37 degrees in a water bath. But there are a couple of times where we have to do, say, a slow hydration, where you add media over a period of almost an hour, drop-wise, a little bit at a time, or you add media directly to the frozen vial instead of thawing it. And so there are a couple of special techniques. We'll cover that a bit later. Most of our items are grown in broth. Some of them are grown in agar plates. We do have amoebas, and I have a picture in here of an amoeba growing on a bacterized plate. Most of my stuff grows in broth, though. And recovery from cryopreservation does tend to vary. Percent recovery is a stat that we do record, but recovery over time is also important because if you have something, for example, with a 90% recovery rate from the frozen state, and it doesn't grow, then it doesn't really matter how good the recovery rate was. But on the other hand, you can have a low recovery percentage to start with and see good recovery over time. So a lot of these different things have to be taken into account. Now, in this next few slides of the presentation, I'd like to cover some specific types of organisms with which I work and point out some of the interesting things about them. So we have the algae, and if you think algae are just green, that's not right. There are different kinds by pigment. To the right there, we have a chlorella, and it is bright green, but below that you see something called Porphyridium purpureum. Purpureum means purple in Latin, and that is a purple alga, and it is very beautiful. I kept it around a lot longer than I needed to just so I could look at it. Usually for algae, we use methanol as a cryoprotectant. The reason I point that out is because it's pretty much the only thing that methanol is used for. As far as I know, you have to use it or they won't survive the frozen state, but then after you thaw them out, you have to make sure to passage them, get some of that methanol out because they don't like to hang out in that after they've been thawed. But that is something kind of unique to algae, I think. You can also see a pigment loss after you thaw your vial, and you might get scared and look at this and say, oh no, why is my algae white? It's supposed to be green, it's the wrong color. Well, I find that if you just leave them be and let them sit in the windowsill and chill out for a week or two, the pigment usually does come off. So that is kind of startling when it first happens, but I think it's actually pretty common. Light source is important too. We find that a lot of our algae do not really like artificial light sources for whatever reason. You wouldn't think there'd be a big difference, but there seems to be quite a difference between artificial and natural. So normally what we do is we put our algae in the windowsill. And this can lead to a couple of problems sometimes. Maybe if you're having a naturally lengthy period of time with overcast days where you're not getting enough sun or something like that. But generally they tend to like the windowsill better. And as I mentioned in an earlier slide, some of these protists can be used for nutritional supplements. Algae are especially worth pointing out because of their many applications. Chlorella has proteins, lipids, vitamins. You can actually buy this as a supplement at the health food store. Brown algae, also known as kelp, is a very good source of iodine, and iodine pills can be made from this. There are red algae that is a source of carrageenan, which is a dairy thickener. So if you've ever eaten sour cream, cream cheese, anything like that, then you've come into contact with this. And finally, of course, green algae can be used as sushi wraps in Japanese food. The next thing I'd like to talk about is bacterized Entamoeba strains, which I mentioned briefly earlier. Why am I talking about this? Well, these are pretty unique in terms of the things that I work with in that they're a special challenge. First of all, they grow in media that has rice starch, and that makes it cloudy. Second of all, they're bacterized, so the bacteria is their food source, so that makes it even cloudier. So it can be difficult to see these small translucent amoebae. So we have some tricks that we kind of use, like using an alternate light source, stuff like that. These grow in the media I just mentioned, PRA 1171, I'm holding a tube of it here, we sell that. And it has the rice starch in the bottom, and then the bacteria is added later. So you need to be able to titrate the amount of rice starch you have. You have to sort of, it's more of an art more than anything else. You have to sort of figure out how much that you need in there. And you really must control your bacterial overgrowth also. If you have too few bacteria, your amoebas are going to die because they won't have anything to eat. But if you have too many bacteria, you will not be able to see what's going on. I like to compare it to taking a bag of marshmallows and throwing it out into a howling blizzard and then trying to pick the marshmallows out of the snow. It's going to be very difficult. So one way that I found that I can visualize these better, and as far as I know, I'm the only person who does this. I haven't seen anybody else try this. So I just decided one day, what happens if I take all the media out of the tube? So the bottom picture here of Amoeba histolytica is the amoebas on the side of the glass, but this is after just about all the media has been removed. They'll be fine for a few minutes as long as you get the media back in there when you're done. So I took the media out, I took this picture, and I put the media back, and it seemed to work. We also have quite a few intracellular parasites among our accessions, and in order to work with these, of course, you must know the basics of cell culture and how to culture the cells. We have a number of cells that we regularly use. As some examples, we have the Vero cell, CCL81. We have this Valve3T3 clone, CCL163. And we have these fibroblasts, HS27, ATCC, CRL1634. And if you look at these pictures, these all show intracellular parasites. The Toxoplasma gondii here on the left, it's green and blue. Those little dots in there, those are the toxoplasmas, and they've been stained with different stains to show the cells versus the parasites. That's an immunofluorescence assay. We take 24 well plates and we seed them with the cell line, and then we infect it with what we're looking at, cryptosporidium or whatever we want to study. And then also you have to be cognizant of mycoplasma testing. We offer a mycoplasma testing service at ATCC, and we avail ourselves of that often because if there's mycoplasma in our product, then downstream it can cause problems with molecular work. So if it's in there, we have ways of getting rid of that, so we do. And I'd just like to point out this neat little picture because this Balamuthia, the brain-eating amoeba in the lower right corner, actually doesn't appear at first glance to be an intracellular parasite, but that's because this spiky amoeba has completely devoured the cell line, and there are just little bits and pieces of it. It looks like a stick of dynamite was thrown in there. We also work with some anaerobic cultures, and here's some of the equipment that we use to do that. So you have incubator chambers. You have various chambers that you can use. There's a square box-like chamber shown here, and then there's a more cylindrical chamber with the top screwed down that people have been using for decades. They still work really well. You take an anaerobic gas generator or a sachet that are commercially available, and you put it into the jar. Now that works for most things, like the Blastocystis hominis seen here. I know it looks like soap bubbles, but that's Blastocystis. It does not like oxygen, but it can tolerate it generally well enough to be worked with on the benchtop for a few minutes. Now, if you have something that is very sensitive to oxygen, can't tolerate it whatsoever, or you're working with large-scale cultures, it is recommended to use an anaerobe chamber that you reach your arms in with the gloves and manipulate things inside the hood, and there's no oxygen at all. And then finally, there's an instrument that you can buy that you just hook it up to this cylinder, and you can put whatever gas mixture you want in there. You just program it into the machine, and in a few minutes it changes out the air and makes the environment that you want. Now let's talk about culture initiation of protists. Some organisms that I've worked with, I haven't worked with all of them, but some that I've worked with, the bacteria and some of the fungal items that I've worked with, they're a little bit tolerant to being thawed, so say you can thaw them more slowly, like over a 5 to 7 minute period, or you can leave them on the bench for a moment when you turn around to get something. A lot of protistology items just will not tolerate that. Some of them will be dying as you're looking at them, so you have to start them in a very timely manner. You know, don't panic. Just start them in a timely fashion. So they have to be stored properly is the first thing. They have to be put immediately into liquid nitrogen. We do have a few things that are freeze-dried, but most of our stuff is stored in the liquid vapor phase of liquid nitrogen. So that's a very cold environment, and as long as they stay in there until you're ready to work with them, they should be all right. Once you take them out, you usually want to thaw them rapidly for 3 minutes or so until they're just thawed. It's 35 degrees centigrade in a water bath, like you see here. And then promptly initiate the culture. Once you've thawed it, put it immediately into the media. Unless it's one of those special methods I talked about earlier, you're just going to put it right into the media and make sure to get the flask or the tube into the proper incubator right away. In addition to that, some protists are even more delicate and they require careful handling, such as gentle pipetting. So if you have a paramecium or a large ciliate, they really don't like to be thawed out, and you're going to have a low recovery percentage to start with. You need to make sure that you pipette those very slowly and gently and kind of treat them with kid gloves. I even had a coworker who used to talk to a protist, and she swore by it. And then finally, of course, the proper incubation conditions need to be observed, not only temperature, but also atmosphere, light source, as we discussed. And then very occasionally, you might see that a shaker is needed. Now, to see what these recovery percentages are and so forth, cell counting is obviously required. And for this, we use a device called a hemocytometer. The diagram you're looking at is a detailed close-up of the business end of the hemocytometer, which is the middle, the counting chamber, where all of the lines converge. And if you look at that, there's a grid in the middle that's 25 squares, and then as the numbers denote, there are these 4 large areas. There are a number of different ways to count your cells. So if you have very large cells and not very many of them, you would do what this slide suggests you do and count the 4 large areas and average them and do a calculation to come up with the answer. If you have very tiny cells like the encephalidozoans I mentioned before that are 2 microns, it's not going to be practical for you to do that. You'd be there all day. So you'd actually zoom into a higher magnification and count the little tiny, each one of those 25 squares in the middle actually has 16 squares in it, and I would be counting those tiny little squares and doing an additional calculation. So it depends on what you've got, working on which technique you're going to use. And then sometimes it's even necessary to estimate, because some organisms really like to clump together. And if you try to get them unstuck, you might cause them damage. So they're just going to be determined to clump together. You can't say, oh, there are 26 cells in that clump. All you can do is estimate, and we do our best. And then finally, counts are not always performed, because in some cases, like where you have to add media to the frozen vial for initiation, as with some of the amoebas, for example, you aren't actually able to count. So we do it when we can, and we have other ways of reporting results, but it's not always done. Now, here are some of the techniques that we use in protistology. I think this is interesting because it's kind of unique to protistology in a way, some of these things. So I've spoken already about slow hydration of freeze-dried cultures and large ciliates like paramecia that are delicate and that you can't afford to just dump the media in there. They'll shock them. You have to very gradually introduce the media. So, for example, for some organisms, you have to start them on a bed of agar with a liquid overlay, and you would add the protist, but you would just have a small amount of the media that they were frozen in, the cryoprotectant medium. And you will gradually, over the course of 40 minutes or so, every 10 minutes, add a mil at a time, drop-wise, to gradually acclimate them. It's like when you're getting into a pool and it's really cold, first you dip your toe in, then you dip your foot in, you're getting gradually. So they have to be handled like that. Also, as I mentioned with the bacterized amoeba, you have to reduce your bacterial density. But here in this picture on the bottom, the Amoeba castellanii, that is, what is depicted there are some cysts, and then the amoebas have come out of the cysts and they're sort of marching across the plate and eating the bacteria as they go. You can sort of watch them day by day devouring this bacteria. You have to make sure that you have grown the bacteria beforehand if needed. It depends. Sometimes it grows well enough if you grow at the same time. It depends on the strain and some other things. But you have to keep an eye on that. Then there's something called picking that I haven't encountered in any other discipline besides protistology. Maybe it exists, but I haven't seen it, where you actually have to pick out individual protist cells from a culture that you have. Like suppose you have a mixed culture, you've gone to a pond, you've collected water from the pond and you see several different things in there. It's almost like an in vitro fertilization thing where you have to go in there with your little tiny pipette and pick out individual cells. Believe it or not, this is an actual technique. I've never done it myself. I don't think you could probably drink a lot of coffee if you were doing that. And then for intracellular parasites growing in cell lines, we talked about this already. You need to make sure that the cell culture is healthy and maintain it. And then also there's in vivo propagation. Now to sort of continue that theme about how protists can be finicky at times, we have some special growth considerations that I would like to mention. So first of all, starvation of free-living protists before freezing. So I had a tetrahymena where they don't like to be thawed. It's a large ciliate, and they do not appreciate being thawed out. They don't want to wake up. So what you do is, in certain cases, you grow the tetrahymena in the media and you just leave them there. And normally when you're growing a culture, you want to make sure it's in a happy state and it likes you. But this one, you leave it just a little bit longer and you make sure the media gets depleted. And it's a little bit unhappy because if it's toughened up like that, when you freeze it, it's more likely to survive the thawing process. There are also differences in medium component lots. So you may see that you work with a particular medium that has a yeast extract from lot 1, 2, 3. And then you work with the next media lot, and you notice it's not growing the organism quite as well. It's the same organism, it's the same procedure, it's the same everything. But they're using yeast lot 4, 5, 6. So things that you wouldn't even think of like that can also make a difference. So if your culture isn't growing, you might want to look into what lot of everything they're using. And then again, I mentioned the type of light sources for photosynthetic organisms, the windowsill versus a 16-hour to 8-hour ratio for a light incubator, for example. And then there's something else that I haven't seen before, depleted media or X factors. So what this is actually growing an organism and letting it grow normally, and then you harvest the organism, and then you take the media it was growing in and you filter sterilize it. And it gives it that particular X factor, whatever, it gives it that special something that enables protists to grow. So that's pretty interesting too. Now, as for preservation, as I mentioned, most protist cultures are stored in liquid nitrogen vapor. They don't necessarily have to be immersed in the liquid nitrogen. They can be in the vapor phase, or they can be immersed. Both work. In order to freeze most of our protists properly, you have to have a slow freezing to about minus 70 degrees Celsius. After that, they can be transferred to liquid nitrogen. You can't just throw them in liquid nitrogen right away. That would be too fast. So we have this cool device, or this nifty device, the CoolCell LX alcohol-free cryopreservation container. And this is just a little chamber where you put your vials. You can see the vials in there. You put the lid on, and you put it in a regular minus 80 freezer, and it causes this slow freezing of about 1 degree a minute until it reaches minus 70, so you can put it in liquid nitrogen. The good thing about this container is that unlike some other kinds of containers for this purpose, it does not have any alcohol in it. Usually you have a chamber with a bunch of isopropanol or something. But with this chamber, you don't even have to worry about that. You don't have to worry about the alcohol going bad or ordering more, disposing of it, or anything. You just put them in here and freeze them. As cryoprotectants, we use a lot of glycerol, we use DMSO, sucrose, and as I already mentioned before, methanol for the algae. And once you thaw something, you have to make sure to dilute it out a little bit so the cryopreservation is not affecting them. And then some protist cultures are delivered freeze-dried or growing in test tubes. Let's talk about QC of protist items. This is my job. I'm a QC biologist, so I test these materials. For the parameters, you're going to follow the product sheet, but also when I test things, I try to follow the bioproduction biologist also. So I follow the product sheet, of course, but I'm also looking at what the bioproduction biologist did so that I can duplicate it. If I can't do what they did, then there might be something wrong with the QC. So I look at both of those things when I'm doing a QC. It's not enough just to grow the protist and say, okay, it's growing, forget about it. You have to look at not only the morphology, or how it appears, but the behavior. And this is one of the interesting parts of working with protists, because they have a wide variety of swimming or gliding or crawling or creeping behaviors. Like to watch an amoeba crawl around through a flask is really neat to watch. But you also have to look at the morphology. So for example, with Giardia, when you start a Giardia, it's supposed to look kind of like a teardrop. It looks like a little sesame seed, 2 little eyes looking at you. And it's supposed to be swimming and flipping and doing all kinds of things. Well, if the Giardia is not happy for whatever reason, we actually describe that as cornflaky because it looks like a cornflake. It's got these ragged edges. It's sort of swimming erratically. It looks like it doesn't know where it's going. It's bumping into everything. So if you see your Giardia with the rough edges like that, that might be a red flag. They may recover, but you should make a note of something like that. And then I also do manual sterility testing for azenate cultures. It's a very comprehensive test. So if I don't think anything's ever gotten past me, if it has, I don't know about it, but if it goes through this battery of tests and doesn't come out with any contaminants, you can be really confident that it's clean. And we do have xenate cultures, as I've already mentioned, but those are also observed for the presence of just the bacteria that you expect to see. For example, if your food source is a rod and you're seeing cocci in there, then obviously something is up. But other than that, we just observe it for the presence of undesirable contaminants. And then there's some specialized testing that has to be done. We have biochemical tests. For example, there's a transgenic T. cruzi we have that reacts with beta-galactosidase as a colorimetric assay. So we have to do a colorimetric assay in that one. We have antibiotic susceptibility testing. So some of our Toxoplasma gondii strains react differently to different antibiotics. And that helps us to classify them and put them in different groups, how they react, whether they're resistant or not. And then there are some special stains, and I'll be talking about those on the next slide. So there are a few stains that we use pretty routinely in protistology. I'd say the most commonly used one is the Ginsa stain. This is used with bloodborne parasites. So on the right side of the picture, you can see Babesia. Those big circles that you see are red blood cells, and then the little rings inside them are the Babesia parasites. And when you stain with Ginsa, you're able to see the contrast quite well between the parasites and the cells, and you can count and calculate percent parasitemia. We also occasionally have to use this acid fast, otherwise known as a Kenyan stain. That bottom picture there is a picture I took a few years ago. It's an Amoeba. It's a pretty interesting organism because it has an endosymbiont that's a mycobacterium. And if you look at this picture, the amoeba is kind of greenish, and then the bacteria inside the vacuoles are these dark purple things, so it really makes them stand out. And then finally, gram stain. We don't normally do gram staining on protistology items for no reason. Where I would really use this is during sterility testing. If I pick up a contaminant in one of my conditions, I'm required to do a gram stain, figure if it's gram positive or gram negative, and then sort of try and figure out what it is and where it came from. Now, we do molecularly authenticate our items, so it is necessary for us sometimes to do DNA extraction and PCR. Whether we're extracting DNA to molecularly authenticate or we're making one of those DNA items like I showed you on the chart earlier, either way we would have to do extraction first. Now there are many commercially available kits. We all have our favorites. Basically you just get one of these kits and you follow the instructions like you're making a cake and you can extract the DNA like that. There are several methods, and we've found over our experience over the years to tweak the existing methods that we have to try and make it better, give us a better yield, or maybe we're having difficulty with some of these problematic organisms. For example, the special techniques section says enzymatic digestion and mechanical disruption. I can tell you one organism that definitely applies to, and that's the Microsporidia. To get DNA from Microsporidia is a little bit of a challenge because they have this hard outer coating. So we use an enzymatic digestion with chitinase, which is an enzyme that'll eat through that, and also mechanical disruption, and usually that is in the form of glass beads. You actually put these tiny 450 nanometer, whatever, glass beads in there, and then you vortex it and it breaks it up and allows us to get into DNA inside. Now the primers we use are universal eukaryotic, 18S, rRNA. There are genus-specific primers against ITS regions of certain organisms in case greater speciation is desired or needed. And there are specific primers for transgenes and genetically modified strains. Now, once we have our PCR product, we can send it for authentication. We use Sanger sequencing to do our authentication, diagram of which is provided here. The 18S nuclear RNA gene is sequenced, and once the sequence is obtained, it can be compared with NCBI and other online databases. So in conclusion, I'd just like to remember a few things about protists to take away from today's talk. They are a highly diverse group of eukaryotes. There is so much diversity, it never gets boring. They have applications in the fields of research, nutrition, and industry. They're extensively studied as models to investigate particular biological phenomena or diseases of humans and animals, and they are fascinating to grow and study. Thank you very much.
Thank you, Katie. In just a few moments, we'll begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Okay, let's start with our first question. Will this presentation be available? So yes, both the slides and the video, recording of the video will be available at www.atcc.org/webinars by Monday. All right, our next question is around passaging protists. So, Katie, are there any hard and fast rules about passage number? For example, how many times can you split them?
I would say there are actually no hard and fast rules. The easy, quick answer is no, because protists are so diverse. There's no way I can just give you a yes or a no on that one. It would certainly depend on the individual protist, but I will say that there's one case that jumps out at me, and that's Cryptosporidium. This is an organism which researchers have not been able to find a reliable way to culture in vitro, because either it doesn't live out its entire life cycle or it senesces after a while. So that's one that's being worked on. So that's one that would occur to me. But the quick answer is no. Some protists, you can just keep passaging them as long as they have a food source and you put them in a new vessel, you can keep going. The only other possible interference I can think of is that eventually you may have some random genetic mutation that would change the protist fundamentally, but that's really only it. You can keep passaging many of them for a long time.
Okay, good, good. Now here's one about growing paramecium. What are the drawbacks to using hay infusion to grow paramecium?
Oh, hay infusion? I can think of two right off the bat. One is that this hay medium is made with this, you can think of it like a tea bag that you put into hot water and it makes tea. So it's basically hay tea. But the thing about it is the particles are very tiny. So they have to filter it something like 10 times to get all these particles out, and sometimes they don't get everything. So you might look in your hay infusion and see itty-bitty particles in the bottom and think it's contamination. I've certainly made that mistake before, where you look at it and you think it's contaminated, it's not. The other thing I'm going to say about hay infusion is that it's very nutrient rich. So there's a seawater formulation and a freshwater formulation. And if you're growing something that's like paramecium that happens to have bacteria with it, as many free-living organisms do, you can end up with overgrowth of the bacteria, so you might need to titrate it out with some seawater or freshwater, depending what kind you're working with.
Okay, good, good. So here's an interesting one. My Euglena look listless. Any recommendations on perking them up?
Well, first and foremost, you should remember that Euglena are photosynthetic. They have a chloroplast and they require light. So if your Euglena don't look particularly happy, you might try moving them to a different spot on the windowsill or maybe using an artificial light source to perk them up. Also, if that doesn't seem to do the trick, what I would recommend for pretty much anything that's not looking happy or like you want it to look is make sure that it's not the cryoprotectant. So if you've just thawed the Euglena and it's got the 10% DMSO or whatever, you might want to passage it to another vessel with less DMSO, see if that wakes it up. You might try refeeding it, or in some cases you can even spin it and concentrate on the medium and refeed that. So there are a number of techniques, but with Euglenas, I would think about the light source first.
Okay, good, good. Now, do protists have to be stored in the vapor phase of liquid nitrogen, or can they be stored at -80 degrees Celsius?
You know, we get asked this question a lot, and it's an important question because the answer I would give is absolutely do not store your protists in -80. I think in some cases you can temporarily put it in the -80, like for a day or two until you get set up or whatever, but if you plan on working with protists, you need to invest in a good liquid nitrogen doer and store them in that liquid vapor phase because many protists will not survive for 2 seconds at -80, and you will not get a good recovery. So I would definitely recommend you don't do that. You store them in liquid nitrogen instead.
Okay. Now, how do I handle frozen ATCC protist cultures upon receipt, and how should they be thawed?
Actually, that's a good follow-up question to what I just said, because I would definitely recommend that you put that protist in liquid nitrogen. There are some that we have that are lyophilized, but most of them belong in liquid nitrogen and arrive as such, frozen. So I would get them in there immediately and then not remove them until you thaw them. And as for thawing, it is important generally with protists to thaw them quickly. So I would recommend putting them in, say, a 35-degree water bath for no longer than 3 minutes or until they're just thawed. Excuse me, and thawing them like that. So you need to make sure they get into that liquid nitrogen quickly, and when you thaw them quickly, and you put them right into the media, and that's how they're happiest.
Okay. Here's a question about the food source. So are there other bacteria in addition to the food that's present when a protistology culture is shipped?
Yeah, that is certainly, pardon me, that is certainly possible, because a lot of these are free-living protists, and some of them have, say, been collected from a pond or a lake. And when you get that collection of water from the pond, you get a whole bunch of stuff in there, and even if you thin it out to certain protists, there can still be more than one bacterium in there. That's certainly possible. The only other thing I'd say about that is, if you're expecting to see a certain kind of bacteria in your protist culture, and you're seeing something, you're supposed to see a rod, for example, and you're seeing cocci instead, that might be a red flag for you to say, okay, maybe there's something going on I didn't expect. But yes, quite often you will see multiple bacterial food sources.
Okay. Here's a couple of questions have actually come in about ATCC's resources for troubleshooting. And I can actually get this one. So
okay.
For troubleshooting, ATCC has a protist culture guide that has information on propagating, freezing, and thawing protists. So it's a good start for suggestions. It's available on our website. And if you're having some real trouble, you can contact our technical service reps. Between all of them, they have hundreds of years of culturing experience, and they can usually suggest a strategy for troubleshooting protist growth issues. Okay, wow, we've got a bunch more questions have come in. This one, I have cryopreserved a number of xenic free-living anaerobic protist cultures in their media with DMSO at cultures that range from 7 to 10%. Upon recovery, the revived cultures often have an estery smell to them even after a number of passages. They smell off, as if the bacterial population has changed. Have you ever seen this before? And if so, were there any steps that you took to get the culture back to normal?
Wow, that's really interesting. Actually, no, I haven't seen that before. To be honest, most of the cultures with which I work are not anaerobic to start with, so the blastocystis and maybe a couple others. But no, I've never run into that particular situation. I'm sorry, I can't shed any light on that. That is interesting, though.
Okay. Here's another one about N. fowleri. Hopefully, first of all, I'm getting this right, but I've tried growing N. fowleri and have not had much luck. How do you grow N. fowleri specifically? What media would you suggest using?
Oh, first of all, you said it correctly. It's N. fowleri, Naegleria fowleri, the brain-eating amoeba, more popularly known. So yes, we do grow that. I don't know if I'm allowed to say specifically what media we use. We grow it in liquid media, whereas some of the other Naeglerias like Gruberi would be on solid media. But one thing I will say about it is that it has a very poor initial recovery rate. It's very typical for me when thawing a Naegleria fowleri to see recovery rates in the single digits. I had one time that was less than 1%. So I think it's important to realize that what's going to recover from that is most of your cells are dead to start with. They just don't like to be thawed. But what you're going to get a good culture out of are those remaining few live cells in there. So I know that you have to be able to see those. We grow it in a couple different liquid medias.
Okay. By the way, it's totally copacetic for you to name the medias as long as you don't name the provider or SKU number.
Okay. These are in-house medias that we use. And I can't, quite honestly, I can't remember off the top of my head which number.
Okay. Okay, so that would be something that I guess I would recommend reaching out to the technical service representatives that I mentioned about for some more insights then. They can probably know how to legally give away the recipe. Okay, this next one's interesting. It's about Leishmania. You mentioned the possibility of freeze drying. Is that possible for Leishmania?
Wow, this is when I wish my coworker was here. She has 10 years of experience working with just Leishmania at Walter Reed. Well, I don't work with too many freeze-dried cultures. I'm not going to say that it's absolutely off the table, but I've certainly never done it with Leishmania, and I honestly don't know what would happen if you tried it. I don't know if we have done any studies like that. We usually don't work with freeze-dried Leishmanias. That's a good question, but I really don't know.
Okay. This is interesting. How do you separate ciliata nuclei to purify micronucleus-specific DNA?
I think that question is too technical for me, Brian. I don't know how to answer that.
Fair enough. Fair enough. All right. Let's see. Pacific University. Okay. Do you have any recommendations for cycling protists that have a digenetic life cycle? Digenetic life cycle. Sorry.
Digenetic. Yeah, I'm not too familiar with that either. I do know that there are ways that you can induce protists to switch states, for example, making them in cyst or exist. So like if you have something that's in a cyst, you can bring it out by giving it nutrients and so forth. And then there are certain bloodstream forms of trypanosomes and things like that that you can induce to be various forms, like different kinds. But I'm not sure exactly how to answer this. I don't have any specific recommendations, no.
Okay. And then let's see, we have one more here. Are there any protists available from ATCC that have their DNA been completely sequenced?
Well, yes, we have a number of them that have been sequenced, yes. So I personally have sent a large number of them off for sequencing myself. So it depends on the protist, but yes, we do have quite a few. And as a matter of fact, that is part of our product line because we sell a number of protist DNA, as seen on a slide earlier. So those have absolutely been sequenced and a number of others that weren't mentioned also.
All right. Let's see. We can wait just a moment or two more to see if anyone has any further questions. We don't have any more coming in, though. But as I mentioned before, the slides and the video will be available on the ATCC website by Monday. So at this time, we will conclude our Q&A session. I'd like to thank Katie for the excellent presentation, and thank you everyone for attending the webinar. One final word. As scientists, we're facing a myriad of challenges and changes due to the COVID-19 pandemic. To help you get back to work, we put together a free kit containing a poster, an infographic, and decals that detail the best practices ATCC scientists use to protect the health of our colleagues and the quality of our research. Order the kit or navigate to this page for other helpful resources keep your lab infection-free. You can navigate to www.atcc.org/backtothelab, all one word, to order the kit or check out the other resources. Thank you again for your time today, and have a great day.